So what does the chapter actually go through? So the chapter starts by talking about biotechnology. As I like to emphasize, there's a lot more to biotechnology than just modern molecular laboratory techniques. When we think biotechnology, at least for me, in my mind, I equate it with taking E. coli, for example, and cloning a gene into E. coli. But there's much more to biotechnology. Biotechnology is something that has been practiced for thousands of years by human civilizations. It is basically taking biological material, organisms, and using those materials and those organisms to do something technological. The example I'd like to give is tapping maple trees to make maple syrup. I mean, that's biotechnology. It is certainly not the equivalent of cloning a gene into E. coli in the laboratory, but it's a technology that has to do with biology. It's biotechnology. Making yogurt, biotechnology. Of course, that's a much more microbiological example, because how do you make yogurt? You make yogurt by having microorganisms that, in fact, grow within milk and change the properties of that milk to give you yogurt. Your book starts in talking about biotechnology, however, by talking about recombinant DNA technology. And this, getting back to my original discussion, is not that unexpected, because these days, although sequencing is so important, 10 and 20 years ago, what was so important in terms of molecular microbiology was cloning genes. And what is cloning genes? Cloning genes is the creation of recombinant DNA. And so cloning genes is still important. Cloning genes, we used to have to clone genes in order to sequence DNA, because in order to sequence DNA, you needed a lot of DNA. Not the case anymore, but it used to be. And so how do you get a lot of DNA? Well, originally what people would have to do is they would have to clone the DNA they're interested in into a microorganism such as E. coli, and then grow large amounts of that microorganism, and then isolate that DNA from the microorganism, and then use that DNA to sequence with. That need for large amounts of DNA to sequence changed a lot, not with modern sequencing techniques, but instead with what's known as polymerase chain reaction. Now, polymerase chain reaction dates back almost as far as my career in science. So I started in grad school in 1984, fall of 1984. And although I'm not 100% certain when PCR technically came on the scene, it wasn't too far into my graduate career that suddenly people were doing PCR, polymerase chain reaction. What's interesting is that I did my undergraduate degree from around the year 1980 through 1984. And the degree was in biochemistry. And the emphasis in biochemistry at the time was not cloning at all. And that is at the undergraduate level. It certainly wasn't PCR, because PCR didn't exist yet. And although sequencing had been invented at that point, that wasn't really much of an emphasis either. So I went from an undergraduate degree where we didn't study cloning to, in fact, a graduate degree, which spanned roughly from 1984 to 1990, where, although I wasn't actually doing it, there was a huge emphasis in my microbiology department on cloning genes, and then there became this emphasis on polymerase chain reaction, and there was this emphasis on sequencing genes. So in a relatively short span of time, suddenly what we know today as these crucial modern molecular techniques suddenly became something that was the center of how people did molecular microbiology. Now, there's a whole set of techniques that's associated with biochemistry, associated with studying individual molecules. And these are the kinds of things that, those are the kinds of things I learned as an undergrad, but that was not the emphasis at all when I got to grad school and people were studying molecular microbiology. And this, in fact, is why I say that when you talk about molecular microbiology, you've got the biochemical aspect, and then you've got what I described as molecular microbiology, which is really all of these various techniques that are responsible for studying especially the proteins and the sequences, the proteins that are found in organisms, the sequences of genes. And these involve the cloning of DNA, and they involve the sequencing of DNA. And then, of course, as I've said, that then was supplanted to a limited but still large extent by just simply DNA sequencing. But nevertheless, the book begins with talking about recombinant DNA technology, and it talks about cloning genes. So the advantages of cloning genes are that you not only generate a lot of DNA, which these days is not nearly as an important aspect, because you've got other ways of doing that, such as PCR, or we don't even really need to do that because of modern sequencing techniques. But cloning DNA is a way of basically producing new gene products, especially new proteins, in a new genetic background. I shouldn't really use the word new proteins, because these are, by and large, although not entirely, proteins that have already existed in some other organism, and we might modify those proteins in the course of cloning and working with them. But we've got these proteins that are being expressed, or these genes that are being expressed, these proteins that are being produced in a new genetic background. And that's what gene cloning gives us. Why do we want to express proteins or express genes? I shouldn't be saying express proteins. Genes are expressed to make proteins. Why do we want to do that? We want to do that for two reasons. The first is so that we can make large amounts of protein. Now, why do we want to make large amounts of proteins? Because if we want to study the functions of those proteins, that is, basically do biochemistry, then it's much easier to do that if we have a large amount of protein. In the past, before gene cloning, to get a large amount of protein, you had to take a large amount of tissue, and you had to take that tissue and you had to grind it up and then isolate the protein you're interested in. Why do we know so much about myoglobin? We know a lot about myoglobin. It's a protein that is basically an oxygen carrier, a storer protein that's found in muscle. And that's because we had available once upon a time large amounts of sperm whale muscle. Sperm whales live by diving deep, 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 deep into the ocean. So they need to bring oxygen along with them. And the way that oxygen is stored in part is in the myoglobin in their muscles. And these muscles, therefore, have a large amount of myoglobin in them. And so if you take sperm whale meat, sperm whale muscle, you can very easily isolate large amounts of myoglobin. That allows you to study myoglobin very easily, because you've got large amounts of protein to analyze. The same thing's true with hemoglobin. Where does hemoglobin come from? It comes from blood. And there's certainly a large potential to tap different individuals for their blood, so that we can come up with large amounts of hemoglobin, or these large amounts of blood from which we can extract the hemoglobin and therefore have large amounts of hemoglobin to study. So we can do biochemical analyses on proteins much more simply, much more powerfully if we've got large amounts of protein. But not all proteins are so easily isolated, purified in large quantities. And what gene cloning gave us is a way to generate large amounts of almost any kind of protein we cared about, so that we could study those proteins biochemically. Why do we study proteins biochemically? We study proteins biochemically to understand what they do and how they do it. And so what I was saying, if you've got a sequence of a community, then you've got sequences of genes in those communities, and you can figure out what the communities are able to do biochemically by what kind of genes are present. How do you know what the genes in the community actually do? based simply upon their sequence? And the answer is because somebody else in a laboratory studied that gene and its gene products biochemically and determined their functions. And at the same time, they also sequenced the gene, so we've got in a database a gene, and the gene is annotated with the function that has been had been previously determined by somebody else biochemically or by the same person biochemically. And then we look for matches between the genes that are in our sequencing set with the genes that are already in the database, and we see a match. We can infer that, in fact, that function that was biochemically determined by somebody else in a laboratory, in fact, may also be present in, for example, the environment that we're studying. So that's the first reason why we clone genes. We clone genes to make lots of protein, and once we made lots of protein, then we can study that protein. And in fact, also, once we've made lots of protein and we're able to purify that protein, we may be able to use that protein in some circumstance, such as medically, where having a large amount of protein may be used, for example, as a drug. Or we might be able to generate a large amount of protein and use it in a laundry detergent as a way of making clothes cleaner. And so all of this becomes possible, this use of proteins in all sorts of different technologies. The use of proteins as drugs becomes much easier when we have the ability to make large amounts of any kind of protein we want, such as through gene cloning. As I say that, I'm reminded of insulin. So it used to be that we had to isolate insulin and purify it in order to provide insulin for type 1 diabetics. But now we use biotechnology to synthesize the insulin. And as a consequence, we've got an unlimited supply of insulin that's produced by microorganisms for us. So that's the first reason why we clone. We clone to generate large amounts of protein. The second reason that we clone, and to a large degree this is harder, is to change the characteristics of the organism that we're cloning into. So if we provide a new gene to an organism, that organism is going to have a new function. And that new function might be useful to us. We hear a lot about genetically modified organisms, or GMO. What is a GMO? A GMO is some kind of organism that some kind of gene has been cloned into. And so we might have genes that provide resistance to that organism to herbicides. And as a consequence, now a plant would not be affected by a chemical that kills plants. And so you could have that plant growing in a field and apply the herbicide to the field, and all the other plants in the field will be killed, but not that plant that has that gene cloned into it that provides resistance to the herbicide. So that's a genetically modified organism. Now, it's harder to make genetically modified organisms because very often we want these genetically modified organisms to be functional somewhere other than in the laboratory. So if we've got something in the laboratory, it doesn't need to be strong enough to survive in nature necessarily to allow us to get something out of it, to make lots of protein, for example. But if we're genetically modifying some kind of organism, we need to set it up so that whatever we put into this organism, and whenever it's expressed, it's not causing so much damage to the organism, the expression of these recombinant genes, these new genes, that the organism is not able to survive wherever it is that we want this organism to survive, such as in a field. Okay, so that's the other reason that we clone genes. We clone genes to genetically modify organisms. And there's some overlap between these two. I mean, obviously, if we're making a protein in an organism, we're modifying genetically that organism to make the protein. But really, the difference is that with GMO, we care about the organism that we've cloned into. Whereas with cloning to make lots of a gene product. What we care about is the gene product more so than the organism itself. We care about the organism staying alive just long enough to make the gene product, but we don't. Our primary emphasis is not on the organism. The primary emphasis is on the product that we've cloned to produce. And with GMO, the primary emphasis is on the organism. We don't care about the fact that these plants make a herbicide resistance protein, a resisting protein. We care about the plant and whatever the plant's going to be making for us, such as corn. Okay? In fact, it's problematic potentially that this corn is genetically modified, that it has this gene that produces herbicide or gives rise to herbicide resistance. But it is not that gene that we're growing the plant for. It is not that gene product that we're growing the plant for. We're growing the plant for something entirely different. So that's really a difference between GMO on the one hand and laboratory biotechnology, where we're simply cloning a gene in order to make large amounts of a gene product. So in any case, what we start out with is basically a discussion on the second page of the chapter of recombinant DNA technology. Because it is important, because GMO is important, because for biochemical analysis, making lots of protein is important. Or we can even study physiology, how these proteins function within the organism we've cloned them in, which is another kind of biochemistry. And all this is based upon the generation of recombinant DNA, the cloning of genes into a new organism.